AKI is associated with high mortality rates, higher than that observed for the most common forms of cancer. The second point from epidemiology is the so-called uremic memory. So patients that develop AKI in the hospital are more prone to develop chronic kidney disease. So these are two reasons for which we need biomarkers. But if you look at biomarkers, nephrology intensivists for AKI are still living in the happy days of the 60s with the man on the moon. By contrast with cardiologists that develop different biomarkers to reduce mortality for acute myocardial infarction, we are still using serum creatinine. But things are moving, the T-bus series are changing, we discovered extracellular planets, and so AKI biomarkers is a topic in the research, so nephrology and intensivists are waking up on this topic. What are the ideal characteristics of an AKI biomarker? We look for early diagnosis, identification of the primitive localization of injury, prediction of duration, severity of injury and outcome, the treatment response monitoring, non-invasive, high statistical power, sensibility, specificity, and obviously for the duration of our hospitals, low cost. Probably this biomarker does not exist, but we know from pathophysiology of tuberous cell injury that after an injury, tuberous cells undergo the loss of polarity, cell death by necrosis or apoptosis. There is a sloughing of viable and dead cells, causing luminal obstruction. And after that, surviving tuberous epithelial cells are able to migrate, they differentiate, proliferate, and reestablish polarity. So biomarker can be useful in this part to identify the early AKI, the early identification of AKI, but also in this part, inducing the regeneration from a good or the so-called maladaptive repair. Indeed, if we have a maladaptive repair, we have an alteration of endothelial cells leading to capillary rarefaction and of tubular epithelial cells leading to the so-called epithelial to mesenchymal transition, so development of CKD due to fibrosis. So we moved from the definition from clinical, biochemical to the molecular diagnosis of AKI. The omics technology entered the world of AKI. We have different biomarkers. I've chosen 3 biomarkers. The first is the kidney injury molecule 1, KIM1, discovered by John Bonvent at Harvard University. This is a transmembrane adhesion protein, normally expressed in proximal tubular epithelium cells, low expression in normal, but is high expression during AKI, and KIM1 is shed into the urine and can be found with different techniques. The most famous biomarker is NGAL, neutrophil gelatinase-associated lipocalin. It's a 25 kilodalton soluble protein belonging to the lipocalin family, characterized by the presence of the hydrophobic pockets able with a high affinity for siderophores. For experiment with gene-reported luciferase for NGAL reported the presence in the thick ascending limb of endless loop in the collecting duct of this gene, slightly in distal tubule and absent in the proximal tubule. So basically, we have NGAL that is produced mainly by liver and neutrophils that are freely filtered by glomeruli and can be reabsorbed by tubular cells for a mingling process. So we have a plasma pool of NGAL and the urine pool of NGAL that is formed by the non-reabsorbed NGAL and the NGAL produced by the thick ascending limb. We have different clinical studies looking at NGAL for nephrotoxicity and also for ischemia reperfusion injury. I reported only 2 studies in the setting of cardiac surgery, so the so-called NGAL score to identify tubular injury. And our group studied NGAL for the early identification of delayed graft function in kidney transplantation. So basically, NGAL is also able, NGAL acts as a growth factor on tubular epithelial cells in acute injury, but NGAL deficiency protects from the progression toward chronic kidney disease. So essentially, the NGAL knockout mice are protected from the development of chronic kidney disease because NGAL is essential in the so-called tubulointerstitial fibrosis due to epithelial mesenchymal transition. The further cell biomarker is obviously cell cycle biomarker, insulin-like growth factor binding protein 7 and tissue inhibitor or metalloproteinase 2, TIM2. So the cell cycle arrest biomarker that have been reported to be with high specificity and sensibility to detect AKI, and probably also this biomarker is essential for AKI to CKD transitions, is as demonstrated again by the John Bonvent group at Harvard Medical School. The cell cycle stop of tubular epithelial cells in the G2M phases lead to the secretion of prefibrotic mediators and progression toward chronic kidney disease. This is the scenario, so we can identify such in acute myocardial infarction the use of different molecules to identify different models of AKI, the early identification, and the possible progression toward chronic kidney disease with adaptive or maladaptive repair. The story of sepsis is very different. We have from John and Rinaldo presentation, the importance of inflammation and the entanglement between sepsis, the systemic inflammation, and tubular cells, in particular with AKI. If you look at NGAL, for instance, in sepsis, we have an increase in presence of systemic inflammation in accordance to procalcitonin. So maybe that during sepsis-associated AKI, we have to evaluate both plasmatic and urine values levels of NGAL. In particular, might be that the peak of urine NGAL is more specific to identify patients with AKI. And also the new cell cycle arrest biomarker can identify AKI in septic patients in a more specific way. If you look to sepsis, we can also identify other selected molecules, such as presepsin and the soluble CD40 ligand, that can predict outcome. Circulating presepsin and CD40 ligand levels are increasing in septic patients and are independently associated with mortality, as shown by these two papers published on critical care. But as we suggested in the Acute Dialysis Quality Initiative work group in Bogota, the sepsis-associated AKI may focus on cells and not only to molecules. In particular, the shift from immunohomeostasis to a maladaptive response to the immune system in the sense of immune activation or immune suppression can be better studied on cells, in particular on monocytes with the expression of HLA-DR. But again, things are moving. We are going from something in the millimolar to nanomolar to picomolar concentration. There is something moving in the research, for instance, the identification of microRNA, small non-coding RNA, able to modulate protein transduction in different target cells. We can identify different microRNA in the circulation of sepsis, of septic patients. And looking at microRNA, what we focused in our lab in the last years was the study of extracellular vesicles. Extracellular vesicles are represented by a family formed from two main families, the exosome, that have an endosome origin and are rather homogeneous population with a size ranging from 30 to 120 nanometers. And they are released by exocytosis through a mechanism dependent on cytoskeleton activation. The second family of extracellular vesicles are the so-called shedding vesicles that are usually larger than exosome, 100 nanometers to 1 micron, and can be released by a membrane-sorted process for different types of activated cells. So basically, the microvesicles are formed by the membrane phospholipid layer containing adhesion molecules and proteins, but can carry different molecules, in particular genetic materials such as messenger RNA and microRNA. Extracellular vesicles have been discovered by three gentlemen that won the Nobel Prize for this, are a mechanism of cell-to-cell communication. And without any doubts, microvesicles are multitasking. So microvesicles are female because you can see the communication between two female microvesicles, very rich, and the poor communication between two male microvesicles is very poor communication. Why microvesicles are multitasking? Because they can stimulate directly target cells, can transfer membrane receptor, can deliver proteins, as I told you before, can induce the epigenetic reprogramming of target cells through the transfer of mRNA and microRNA. And obviously, microvesicles can be a biomarker. So the first question regarding microvesicles, are urine extracellular vesicles biomarker of AKI? The answer comes from the barley. Barley is a very clever plant. After dehydration or infection, induce the release of extracellular vesicles or microvesicles. And also the kidney. I know that the kidney looks like a bean and not a barley, but behaves like a barley. So in condition of sepsis or ischemia reperfusion injury, may release some extracellular vesicles in a condition of stress. And large-scale proteomics and phosphoproteomics of urinary exosome identify more than 1,400 proteins. In particular, the phetuin A has been identified in different experimental models, as also in humans, as present in the microvesicle fraction in the urine as potential biomarker of AKI. These are data from our lab. Also, the NGAL, a part of the NGAL present in the urine is not free, but is bound on the surface of extracellular vesicles. And extracellular vesicles are also positive for messenger RNA, for instance, encoding for NGAL. So another important thing for urine microvesicles as biomarker is the identification not only of protein, but also of nucleic acids between inside the vesicles. In particular, you can see the expression of different microRNA profiles may be predictive of allograft status in kidney transplantation. This is something we also studied in our lab. But if you shift from urine to plasma, extracellular vesicles can be identified in the bloodstream of normal people, of healthy people, and are mainly derived from platelets and mononuclear cells. And they contain, obviously, different types of RNAs, including microRNAs. So the question here is if inflammation during sepsis can lead activated cells, activated leukocytes, platelets, and endothelial cells to release a different pattern of extracellular vesicles and microRNAs. We look for as biomarker, obviously, at microvesicles in the plasma of septic patients, but our feeling is that microvesicles are not only biomarkers, but may have a pathogenic role in the mechanism of sepsis-associated AKI. This is the study in collaboration here with the International Research Institute in Vicenza, Renal Research Institute in Vicenza. This instrument is called NanoSite and allows the determination of concentration and size of microvesicles in body fluid. You can see the different type of microvesicles present in the plasma of healthy or septic people. And the concentration of microvesicles in the plasma is associated with a poor outcome and with an increasing number of organ failure. These microvesicles in the plasma by flow cytometry express different adhesion molecules of the integrin family, HLA-1 and 2 class antigen, and protein involved in inflammation and apoptosis, such as CD40 ligand and FAS ligand. The RNA profiling of these cells is under investigation. But this is true not only in septic patients, but also in experimental models. We can see that after LPS infusion in mice, second ligation and partner in rats, we have an increased concentration of circulating plasma microvesicles. And interestingly, if you take these vesicles from aseptic animals and inject them in healthy animals, you can recreate sepsis-associated AKI. And you can see the localization of microvesicles labeled with a red fluorescent dye in the kidney of healthy mice. And the same thing in vitro on tubular epithelial cells. And when we look at the histology, histology is very poor, as Fernando Bellomo showed, but there are some alteration of microcirculation, and microvesicles taken from septic animals induce a release of urine volume and an increase of serum creatinine and blood urine nitrogen. So we have a microcirculatory derangement. If you look at the tubular cells by immunohistochemistry, we can see that the microvesicles from septic animals induce a reduction of aquaporin 2, a reduction of PGC1 alpha, that is a protein involved in mitochondrial biogenesis. It is essential for the regeneration of tubular cells in sepsis-associated AKI, and an increase in NGAL. Another interesting point is that if you treat with RNAs, if you pre-treat with RNAs the microvesicles, RNase is able to be internalized into the vesicle, destroying all the RNA subtypes. The detrimental effect of microvesicles are not observed. So, coming to conclusion, we think that biomarkers and AKI may lead to personalized therapies. For the first time, to identify patients more prone to develop AKI and progression toward kidney disease. And we can also use biomarkers to see what we are, if you are doing something good with kidney regeneration or something wrong therapy, wrong timing. So identifying not only the outcome, but the progression toward AKI to CKD. And this is what we also proposed in acute dialysis quality initiative in Virginia. We think that biomarker can identify also some new therapeutic targets to drive the kidney toward repair. And so we are still dreaming for sepsis and AKI biomarker. Thank you.